This lecture is got a lot to it, meaning there are several key concepts that carry us through the rest of the semester. Specifically, I mentioned this, we're probably not going to get to too much of it today, but signal transduction pathways is such a key component because it teaches you the ways in which cells will tell each other what to do during the development process. How they're able to say, well, you're becoming skin, you're becoming bone, you're becoming this and that. It's the way in which they communicate with one another. But before we can get to that, we first have to look at how cells know what they're supposed to attach to. We'll talk about potency, their pluripotency and multipotency, and that really comes down to what we call competence and specification, the ability to respond to a signal and the stepwise process that they take to take on their ultimate fate. So these are the three main factors that this lecture addresses, which all have to do with morphogenesis as well as differentiation. The first part is mostly morphogenesis, but there is a relationship between morphogenesis and these others as far as differentiation goes. What I mean by that is morphogenesis, as we've talked about, is the movement of cells in relation to one another and how they start setting up the body plan. And most of that comes down to cell adhesion. However, one of the ways in which they're able to alter and change their adhesion to move around comes down to their ability to respond to these signals via the signal transduction pathway. So this is both morphogenesis, but when we get more into these, this is going to be more competence and specification. Signal transduction pathways is going to delve more into the actual differentiation process. Remember how I made a big distinction between morphogenesis and differentiation. So, cell adhesion. Ultimately, in multicellular organisms, the relationship that cells have with one another determines what their fate will eventually become. So morphogenesis plays a key role because if they're in a different part of the embryo than they should be, they're going to be under a different set of signals in relation to those other cells and take on a different fate. So position matters substantially. So the question becomes, how do the cells know where they're supposed to be? What are the factors that tell these cells to fold inward and move around this way and these to stay where they're at and the like? Well, one of the major proteins are these cell surface molecules called cadherins, or calcium-dependent adherent proteins. So that's where the word cadherins comes from. Is these are calcium-dependent proteins on the surface of the cells that they interact with other cell membranes with their cadherins that play a critical role. Now, not all cadherins necessarily form connections with one another. There are cadherins that have a high affinity for one another, and then there's other receptors that actually repel one another. So it's not always about just holding hands with all the other cells. It's also about, get away from me, I don't want to be near you. That ultimately allows these cells to be organized into the critical pattern to become these organs and tissues. So the two major properties in this morphogenesis process are not only the type of cadherins, which is what we call quality, but the amount matters as well. So the more that they have on their surface, the stronger the adhesion to each other will be, and this aids in the process because, as I'll show in a couple of slides here, that they've done some experimental studies where they've taken different ectoderm, mesoderm, and endoderm layers, and they've mixed the cells up and then let them kind of sort themselves out, and they always sort themselves out in certain patterns based upon the quantity and quality of the cadherins. It just naturally occurs based upon what's being expressed in the surface of these cells. So there are a number of different things that they did. They took presumptive ectoderm, or these epidermal cells, and they took neural plate cells, which are a little bit further down the line. They've gone through a little bit more of the specification process, where they're not just ectoderm, they're actually neural ectoderm, where they're going to become the neural tissue, such as the spinal column or the brain. Well, then they break them up. Now, one of the ways in which you can break up cells, when you take cell culture, one of the ways in which we cause cells to dissociate from each other is to use a protease called trypsin. Trypsin actually comes in and just kind of indiscriminately starts cutting proteins off, especially cadherins, especially proteins on the surface of cells. So they use this trypsin to deaggregate and cause all of these cells to just become individual cells. And then the cells are still capable of reestablishing those proteins, and so they just make more of them. They've got their kind of surfaces cleaned off, they make more of them, they reestablish that. So over time, what happens is the cells start reaggregating according to their affinity for one another based upon the type of cadherin. So there are many types of cadherins, and only those that have the same type typically interact with one another and form these epithelial layers in conjunction with one another. But another question becomes, well, how come the neural ectoderm always comes in closer than the epidermal cells, which will become the skin? Why do they always come? Why not have the neural cells on the outside and the epidermal cells on the inside? What do you think? Quantity. So here, the quality, the type of cadherins tells you what cells are going to associate with. The quantity typically tells the cells what layer they're going to be in. So the ones that have more of the cadherins typically become the ones that are the inner cells. And that's one of the things that they've shown, is that this is how they're able to separate from one another, is that the ones that have fewer connections make room for the ones with stronger connections for the inside, and so on and so forth. And that's how they start forming these layers, is by quality and quantity. So here's just a series of different experiments that they did with all sorts of different layers, from epidermis to mesoderm, mesoderm and endoderm, all three, epidermis, mesoderm, and endoderm. What's interesting about these two is endoderm and mesoderm tend to have the same quantity, which normally endoderm would be on the inside. But then that begs the question, well, why, when you have epidermis, mesoderm, and endoderm all together, that the endoderm will always go to the inside? What do you think? Why would, in this scenario, when you just have endoderm and mesoderm together, the endoderm is supposed to go as the most innermost layer, but it pretty much comes out here. So why, when you add the epidermis, that the endoderm will now go towards the middle? Differential affinity and repulsion to other tissues. So the endoderm not only likes to be with itself and be close to mesoderm, but it's actually repelled by the epidermis, which means that it will come inward rather than, say, be on the outside. So the addition of this illustrates the fact that when you start putting multiple layers together, you not only get attraction with itself and possible secondary affinities to the germ layer that it's normally used to, but it also repels from others as well, so that they'll always organize themselves in that fashion. So it doesn't come, as I mentioned, it's not just attraction to others of its type, there's also repulsive elements that push it away from other germ layers. And you see this in all of the development of any model organism, that you always have ectoderm on the surface, mesoderm inward, and endoderm even further inward, because their ultimate fates need to be in those respective areas in relationship to one another. We don't just use trypsin to deaggregate the cells from, say, a matrix. We also have to use a chelator. You guys remember what a chelator is? It binds essentially ions, and one of the ions that you have to get rid of is calcium. So we use a chelator called EDTA to help in the process of getting rid of the cell-cell interactions that's due to these cadherins. That's one of the things that we learn how to do in cell culture. So without calcium, if you were to put a chelator in here and absorb those ions, even though you had the presence of these, they wouldn't connect with one another because they are calcium dependent. That's where they get their name from. Now, one of the other things that will become important later on is in such close proximity, there are other proteins that can be in the membrane that it's necessary for the cells to interact with one another in this way so that those proteins can interact and thereby cause different changes to occur in the genomic transcription and translation of proteins. As we mentioned last time, that's really what we're interested in, is knowing what genes get turned on and transcribed into RNA and translated into protein, because these are not static, they're dynamic. The cells can, over time, as they go through the differentiation process, change their cadherins. And that's another reason why at one point they might be attractive and another point they might be repulsive. So just because at this point in time these cells are adhering to one another, there could be some cell-cell interactions here that cause these cells to create a different type of cell adhesion molecule that cause them to aggregate another way. So these are dependent upon what stage the cell is at, and that gets into the differentiation process. So morphogenesis is the movement of the cells around one another, but they can alter how they move as they progress through their differentiation into their tissue or cell type. So here's just a couple of examples of cadherins, and we usually name them based upon where we find them, especially in early development. So we say E cadherins for the early embryonic mammalian cells that will be part of the epithelial line. So that's where the E comes from. P cadherins are cells that are extra-embryonic and typically become the placenta. N cadherins for the nervous system. R cadherins for the retina. B cadherins for other neural structures. We've got, names for all of them. So these are just a sample of some of the types of cadherins, and there are many more than this. But these different versions ultimately determine what association the cells are going to have with one another on the surface. Okay?